Next major topic that we are going to start, it is changing genes, that is site directed mutagenesis. पहले हम फोकस करेंगे कि म्यूटेशन या म्यूटाजेनेसिस का जो प्रोसेस है ये क्या होता है. So actually mutagenesis is a process by which we can change or alter the genomic DNA of an organism, and this change it is relatively stable and it is also called as a mutation. Now mutagenesis, it can takes place in different ways. It can take place naturally, and even another way is that when living organisms they are exposed to certain mutagenic agents, it can also induce changes in DNA sequence. And nowadays the basic you can say that the tool to study the functions of different genes, we can also induce the process of mutagenesis in the laboratory. अगर ये प्रोसेस नेचुरली हो तो इसकी वजह से लिविंग ऑर्गेनिजम्स में बहुत सारी प्रॉब्लम्स हो सकती हैं, लाइक इट कैन इंड्यूस द कैंसर and different type of diseases. But overall, biologists have the viewpoint that mutations or the process of mutagenesis, naturally it is the prime force during the course of or the process of evolution. के इर्तिकाई अमल में जो जीन्स का चेंज होना है विद द पैसेज ऑफ टाइम, ये इर्तिकाई अमल की बुनियाद है. But overall we can say that the mutagenesis it can takes place under natural conditions and may be triggered by certain mutagenic agent and can be induced under laboratory condition. And this process of mutagenesis, it was first time reported around 1927 by Hermann Müller while conducting experiments with fruit flies. And he first time detected the phenotype related to the mutation when he irradiated fruit fly with radiation like the X-rays. And then later on, it was also proved that certain other environmental factors as well as pollutants, they can also induce changes in DNA structure. And first, while discussing different causes or mechanism of mutagenesis, briefly we can first focus that what is the difference between DNA damage and mutations. जहाँ तक DNA डैमेज का ताल्लुक है तो इसमें जो स्ट्रक्चर है अगर DNA का वो जब डैमेज होता है तो अल्टर होता है तो उसकी जो DNA की रेप्लिकेशन है ड्यू टू डैमेज वो स्टॉप हो जाती है. तो हम ये कह सकते हैं कि लाइक अगर कोई सेल है या माइक्रोब है तो डैमेज की वजह से जो DNA है या हेरेडिटरी मटेरियल है वो नेक्स्ट जनरेशन में ट्रांसफ़र नहीं होगा. But on the other hand, in case of mutations that may result in change in sequence of the bases, either as a result of addition or deletion of certain bases, तो उसकी वजह से क्या होता है कि म्यूटेशन जो है इसमें DNA की जो रेप्लिकेशन है ये स्टॉप नहीं होती. Rather you can say that this change it is heritable and can be transferred from one generation to the next generation. So this is the difference between the damage of the DNA and the mutations. Now what may be the different mechanisms by which mutations can be induced? Mutations can be induced endogenously or can be triggered by different environmental interference or exposure. As far as the endogenous causes are concerned, they may be hydrolysis of the bonds of bases. For example, in case of purine bases, thousands of bases may be present within a short DNA segment, and naturally the bonds between the bases it can be broken as a result of depurination. So purine base bond it can be damaged. or purine base, it can be removed. And cell has some natural mechanisms where the DNA bases, they can be again replaced with guanine or related purine bases. But on the other hand, if natural mechanisms of repair, if they are not working, then other bases like adenine, they may be incorporated, and in this way the natural process of mutation, it can start. So the hydrolysis, it is also one of the reason that bonds may be broken as a result of hydrolysis, and different bases, they can be incorporated. Another method that can be carried out endogenously, or even it can be triggered by certain chemicals or drugs in vivo, it is the modification of the bases. Now, modification, it may be as a result of methylation or glycosidic bonds of sugars, they can be modified. और इसके अलावा जो process है, like alkylation का जो process है, it is also very important. जो alkylation है, ये किसी ड्रग के यूज करने से भी हो सकता है, या किसी केमिकल म्यूटाजन की वजह से भी हो सकता है. तो इसके साइड इफेक्ट्स ये हैं कि जब एल्काइलेशन होती है डीएनए बेसिस की, तो इसमें एक्सटेंसिव क्रॉस लिंकिंग हो जाती है, इधर इंट्रा स्ट्रैंड और बिटवीन अपोजिट इंटर स्ट्रैंड्स. तो उसकी वजह से, क्रॉस लिंकिंग की वजह से डीएनए की रेप्लिकेशन जो है वो स्टॉप हो जाती है. तो इस तरह से आप ये कह सकते हैं कि इट इज़ अ सॉर्ट ऑफ डीएनए डैमेज. तो इसके अलावा जो और डिफरेंट कॉजेज हैं, लाइक एक्सपोजर टू यूवी रेडिएशन नेचुरली, इट इज़ मे बी इन्वायरमेंटल फैक्टर, कि जब यूवी को एक्सपोज होता है डीएनए, तो उसकी वजह से जो दो एडजेसेंट थाइमीन रेजिड्यूज हैं, बजाय कि वो अपोजिट स्ट्रैंड के एडेनिन से पेयर करें, वो दे कैन पेयर बिटवीन दैम, and in this way the thymine dimers are produced that can also hinder the DNA replication. Additionally, some intercalating agents, if they are introduced in the laboratory, like the addition of ethidium bromide, for example, if it is there in the bacterial culture, it can preferentially target the plasmid DNA. So plasmid, it is lost during the subsequent cell division, and same problem may be caused by some other mutagens like the proflavin. So these are different mutagenic agents that can induce changes in DNA structure. But the DNA changes or mutations, they can also be induced under laboratory conditions to study the functions of different genes, and this specific subject we can discuss under the heading site-directed mutagenesis. And there are different laboratory techniques where we can use this process of site-directed mutagenesis. For example, three main types are used, like asset mutagenesis. Next one, it is primer extension, and some protocols or procedures that are based on polymerase chain reaction or PCR.